Welcome. We are live at NASA today from two locations. We're here at the Goddard Space Flight Center, and with me I have Dr. Paul Mahaffey, who is actually our director of the Solar System Exploration Division, and he's also the principal investigator for the SAM instrument, which we're going to be talking a lot today. And I also have Jennifer Eigenbrode, who has one of the coolest titles I know of at NASA, which is actually an astrobiologist. And we're also here at another location today as well, NASA's Jet Propulsion Laboratory in California. And here we have Dr. Chris Webster, who is a senior research scientist at JPL. And we also have Ashwin Basavada, who is the project scientist for the Mars Science Laboratory. My name is Michelle Thaller. I'm an astronomer here at NASA. And one of the things I'm going to say right off the bat today is that none of us live in a vacuum. We all have our own social media accounts. And we're sort of aware of the interest that this has generated. So I'm going to tell you right now, what we're not announcing today is the detection of life. We're not going to be talking about that. What we are talking about are some really exciting new results that have to do with organic molecules. Now, organic molecules are what we understand of as the building blocks of life. And even if we're not announcing the detection of life today, I think that the discoveries we're going to be talking about are a really important next step into finding out whether Mars was once habitable or might even still be habitable. So what I'm going to do now is actually lead it off with Paul Mahaffey, who's going to talk to us a bit about what we know and what we don't know about Mars. So Paul, straight up, what have we found?
Really exciting times. I'll reinforce what you said. I mean, organic compounds on Earth just so much bear the imprint of life that it's very natural for us to equate finding organics on Mars with finding life. But that's not the bottom line of what we're trying to get out here. The bottom line is that we've greatly expanded our search for organic compounds, which ultimately are fundamental to our search for life. And it's really interesting, kind of two complementary results. One result is organics from billion-year-old rocks that got trapped in the rocks in an ancient lake billions of years ago. And the second really is the simplest organic, methane in the atmosphere. And those two results, Jen Eigenbrode and Chris Webster, the lead author on these papers, are going to talk about. But let me just set a little bit of the context. It was kind of August 2012, more than 5 years ago. We came barreling into Mars, and there was that exciting 7 minutes of terror. And we landed safely on the surface thanks to the great engineering team at JPL that got us there. But the objective of the mission really was to explore a habitable environment on Mars. And right off the bat, we just found incredible things. We found clays that were formed by water. The rover Curiosity is just extremely capable. It takes stereo pictures. It measures the elemental composition of the rocks. It does the mineralogy. And what we've always done, even though we found many things with the experiment we're going to talk about today, which is SAM, the Sample Analysis at Mars experiment, we essentially found out very early that this was a habitable environment. Water had been there for a very, very long time. With our SAM experiment, we found some interesting things. For example, how old the rocks were, how long they had been exposed to cosmic radiation. Measurements of light versus heavy elements told us about how the atmosphere had escaped over billions of years. But we've kept going on this search for organics, and we had found some methane before. We had found some simple organic molecules before, but we greatly expanded the search with the results we're going to talk about today. So it's really exciting.
So Paul's just mentioned some of the results we're talking about today, and one of the things that's really important is you can be involved in our discussion. So throughout the broadcast today, you'll see me looking down at my iPad. We're taking live questions. So if you're on social media, you can use hashtag AskNASA. Or if you're joining us on Facebook, just put your questions in the Facebook comments, and we'll get to as many of them as we possibly can. And we're also going to be taking questions live from the media a little bit later in the show. So I'll actually be here live looking at your questions, and I can give them to whatever scientist is probably the most appropriate. So now we're going to go to Jen. So Jennifer, tell us a bit about, why is it significant that we found organics on Mars?
We found organic molecules in rocks from an ancient lake bed. Those organic molecules could have come from life. We don't know that there was ever life on Mars. The organic molecules that we found are not specifically evidence of life because there are other sources of making those molecules, including things that are non-biological in nature, things like meteorites, or even rock processes. We can attribute geology all by itself without life to making organic molecules. And the information that we have doesn't tell us which source is responsible for what we have.
Well, thank you. So another one of the major detections and discoveries we're talking about today involves methane on Mars. And methane is something that we think of as natural gas. It's a simple organic molecule. And I'm going to talk to Chris Webster at JPL right now. So Chris was actually instrumental in the discovery before of methane spikes. The level of methane went up very dramatically on Mars. So Chris, you've reported before on these methane spikes. Can you tell us a bit about what today is different and noteworthy about methane on Mars?
Well, Michelle, as you know, every chapter in the story of the methane on Mars has been a surprise. From some time ago, there were reports of plumes in the atmosphere of methane. There were reports of patches when orbiters looked at it, and spikes that we reported, as you just mentioned. Every one of them a surprise, but problematic in the sense, frustrating, because none of them were repeatable in time and space. They seemed to show that methane was misbehaving. It was sporadic, or almost random pulses and patches were showing up. So today we're announcing a discovery of a repeatable, identifiable seasonal pattern in the methane measurements. And we can look here at the graphic and we can see that. It's in the lower background level, because most of the time we're not looking at spikes, and we see this low background level. You can see from the winter to the summer this growth. And the big surprise too is not only have we got this wonderful repeatability, but the seasonal cycle changes by a factor of 3. That's a huge change, completely unexpected. And what it does, it gives us a key to unlocking the mysteries associated with Mars methane, because now we have something to test our models and our understanding against. And we'll hear a little more about that later.
So one of the things you're hearing a lot about today is the term organic, and this is something that a lot of people are familiar with in many different ways. So I'm going to talk to Jennifer Eigenbrod. So Jennifer, tell us a bit about what do we mean by organic, and tell us about what is so significant about finding organics on Mars.
Right. So organic molecules to a chemist are simply molecules formed of carbon and hydrogen. Sometimes there's other things that go in that, such as sulfur, nitrogen, oxygen, phosphorus. However, to a chemist, it's just that type of a structure. What we're not talking about is molecules that specifically come from life, and we're really not referring to the organic stuff that you find at the grocery store, which is that pesticide-free produce that we get. They're very different things.
So it seems to be kind of amazing. I mean, this is one of the engineering feats of the Mars Curiosity rover, is that we're finding molecules. We have a chemical laboratory on another planet. Can you talk to us about how we identify these molecules on another planet all the way on Mars?
Sure. So the Curiosity rover drills into rock layers. And when it does, it produces a rock powder, and that rock powder gets put into the SAM oven and heated. And when it's heated, it produces gases. They get whiffed off into the mass spectrometer. Here you can see the gases coming through some tubes, and they enter into this chamber where electrons ionize that material. As a result, you have molecules that are split into tiny little pieces, and the mass spectrometer can then identify what those different pieces are, and we can put them back together to understand what the original molecule was. Now, there's another feature of SAM. That is, it has a gas chromatograph. And the gas chromatograph looks a little bit like this. It's a really, really long tube, and it has a hole in it that's about the width of a human hair. Now, the column allows molecules to go through it, and they go down this long, long tube, and when they come out the other side, they come out one by one. They're separated. And when that happens, they go into the mass spectrometer, and we can identify individual molecules. Now, the GCs on SAM were built by our French colleagues from the French National Center for Scientific Research. And it was because of these GCs that we were able to identify certain molecules that we are reporting on today.
And in fact, actually, we have one of our first questions from social media. This is from John Vandelar. We were talking about drilling into the surface of Mars with the arm of SAM. How far in do we go? How far deep into the surface?
5 centimeters. That's about as far as we can go. Yeah. 5 centimeters.
We'll talk a little bit more, John, about why that's significant. There may be even more interesting things farther down. So the thing that really amazes me, and I remember very well the landing of the Curiosity rover as one of the best nights, early mornings of my life. When you showed me that tiny gas chromatograph coil, that had to actually land on Mars. At one point going through the atmosphere, there was a big snap of G-forces, like up to 10 times the force of gravity. We actually land these incredibly sensitive, sophisticated instruments on Mars. And I want to talk a little bit more about that. So we're going to go to our friend Ashwin at JPL. So tell us where you are and tell us a bit about the Curiosity rover, the larger rover.
Hi there. Yeah, probably wondering what's behind me. And what you see behind me is actually the test model of the Curiosity rover. It's a twin, basically, that we use for testing here on Earth. The one on Mars is in a place called Gale Crater. We sent it there. It's about a 100-mile diameter hole in the ground that formed when a giant impact occurred on Mars about 4 billion years ago. But what drew us to this crater is the mountain that you see in the middle of it. This mountain wasn't there when the crater formed. In fact, it formed as sediment was carried in by rivers and streams and then deposited into this giant lake that filled the crater. And as that sediment settled out from the water, it built up the layers that then made up the mountain. Each layer of this mountain is younger than the one below it. So by climbing this mountain with the rover, we can read the ancient history of Mars. And climbing is exactly what we've been doing throughout most of Curiosity's mission. We're now 1,000 feet above the crater floor. That's as high as some of the skyscrapers in downtown Los Angeles. And we get this amazing view. And so the progress has just been to continually drill each layer of this mountain. And the samples that Jen described were taken from the lowest part of the mountain about a few years ago in 2015. And it's taken us a few years to get an understanding of these results so that we can describe them to you today.
And Ashwin, there was a reason that Gale Crater was selected. There was something really fascinating about the rocks, some evidence of water. Can you tell us a bit about why this site was selected?
Sure. I mean, we had some hints that water was involved in interacting with these rocks because of the orbiters that have been at Mars before us. So, for example, there's the Mars Reconnaissance Orbiter and there's Mars Express, and both of those instruments had both of those orbiters had instruments capable of detecting clay minerals at the site. So we know that some of the layers in the mountain are made of clay minerals, which means that water had interacted with them. We weren't sure, though, if rivers and lakes actually existed, and Curiosity has now shown that lakes existed for a long time, probably hundreds of thousands, if not millions of years.
And actually, I have a question from social media. So Jean Pascal from Facebook says, tell us more about the tweak and the repairs made to the arm. There was some trouble recently with the drilling arm. And space is hard. You're all the way on Mars. Tell us how you actually fixed this from millions of miles away.
Yeah, this is really worth a press conference of its own. It's just sort of coincidence that we have this amazing week to tell you not only about the success of the drill, but about these wonderful results from the last few years of exploration as well. In late 2016, we realized that one of the main motors of the drill was no longer functioning reliably. This basically meant the drill was dead, and a big part of what we could accomplish on Mars, we'd no longer be able to do. It's not just that we fixed something or tweaked something, we had to invent a whole new way of drilling. And that's just a remarkable story that really I have to give all the credit to the engineers at JPL who did that work. So for about a year and a half, they tried to fix the motor, then invent this entire new way of drilling that didn't use that motor any longer. And just this week, we have achieved success. We've now successfully drilled and analyzed samples with our laboratories once again.
So we're talking about drilling down and analyzing these samples. And for those of you that are a fan of organic chemistry, we're actually going to dive into the details just a little bit right now. So Jen, tell me the details about what was detected.
Right, so we detected a variety of molecules, including some that have carbons linked in a ring structure, such as benzene, and others that include carbon chains. And here's an example, propane. Now, these are just representative of molecules, small little bits and pieces of molecules, but because we see these coming off of the sample at high temperatures, what they're really telling us is that they're part of something larger, a macromolecule. And this is just an example of something we find on Earth. We call it kerogen. Now, we find examples of kerogen on Earth in things like coal and black shale, these two things. Right? And we also find them in meteorites. So they're common. This tells us that there is organic material in the rock in a different form than what Sam detected. And what it means is that it was in a robust form. This form of organic matter resistant to changes. Now, the other thing that we found was thiophene. And this is an example of some of the data we'd get from Sam. We can actually tell after the molecules go through the GC column the actual structure. We use this information to figure out this type of structure. So here we have 4 carbons and a sulfur. Now sulfur looks like this when it's in rock form. But in the sample, the sulfur is probably in the organic molecules themselves. And that's important because sulfur actually is the material that can help bind all these small pieces of organic molecules together into something really big like that large molecule I showed you. And sulfur helps to resist oxidation. That's really important for our discovery because we are looking at ancient rocks that are 3.5 billion years old, and we found organic materials inside them. However, we drilled in the top 5 centimeters. And in that top 5 centimeters, the surface is exposed to a lot of ionizing radiation, and that generates free radicals and oxidants. And all of those can degrade organic materials. And so that rock has been sitting at the surface for an extended period of time. We're talking tens of thousands to hundreds of thousands of years. And that's a long time for all these changes to happen. There were a lot of people who thought we weren't going to find all the organic molecules that we did. For instance, Viking started this quest back in 1976, looking for organic molecules. Curiosity has continued that quest. And now we have a diverse set of molecules that we can now start to understand a little bit more about how this material is preserved and where else we might look to get more.
This is a pretty amazing thing. I mean, working on Mars is very hard. I mean, a lot of people know that Mars has almost no atmosphere compared to the Earth, about 1% the atmospheric pressure of the Earth. It has very high radiation levels because Mars isn't protected the way the Earth is by an active magnetic field as well as a thick atmosphere.
That's right.
So we have this laboratory working on Mars. And so the radiation from the sun gets about how far down into the soil?
It's coming from the sun, but it's also coming from the galaxy.
Right.
And it can extend about 1.5 meters down, maybe even a little bit more. It depends on what model you use. But there are places you can go deeper down that we might be able to tap into molecules that aren't disrupted by the radiation itself. And the ExoMars rover is a perfect example. It's going to drill 2 meters deep. This is the European Space Agency's ExoMars rover. And it's going to drill 2 meters deep, and it will have the opportunity to examine materials at the surface and on the way down to see if there's comparisons. We'll learn how important it is to where we have to refine our analyses, where we'll understand the preservation issue and how it affects things at the surface as compared to things further down. But most importantly, if there are biosignatures inside that organic material or associated with it, we're going to have a better chance of understanding that if we get to materials that are not exposed to all this radiation.
That's one of the things today. We're finding some very interesting chemistry right up on the surface of Mars, and it's a promise of even more interesting chemistry to come if we can get a little bit deeper down underneath that. So, we're looking at Gale Crater, so how did these organics get here? I mean, we're finding these organic molecules now on the floor of Gale Crater. How'd they get there?
So what we're looking at here are a lot of layers that were put down in an ancient lake. And as previously we discussed, that lake had been there for a long period of time, hundreds of thousands, maybe even millions of years, we're not sure. But those lake sediments are for a gigantic lake. Organic materials can come into that lake through rivers. They could be wind-blown in, or they can be formed in the actual lake itself by things like biology. Now, there's 3 different sources of organic materials, too. One of them is the biology. One of them could be meteorites that are infalling into the surface and being directly deposited. And the other one is a rock forming processes that might generate organic material all on their own, and they might be the ones that are being brought in by rivers. We don't have enough information from the observations we've made to tell what the source is and how it got in there.
So we found this really good location to look for organics on Mars, and we're also going to talk now about the seasonal methane. So, I mean, are your discoveries about the sorts of organics and the site, is that all related to Chris's stuff about the seasonality of methane?
Yes and no. If you take the organic detection in the ancient rocks, and you just generalize it, we have found organic material in the subsurface. And that might be representative of organic material throughout a grander area. And that is carbon that could go through various processes and make its way to the surface and turn into things like methane and then seep into the atmosphere. Now, the detection that Chris is going to talk about is about modern day methane, as opposed to ancient signals, what I discovered in the rocks.
All right, so with that, we're going to go over to Chris Webster back at JPL. And so Chris, tell me a bit about the seasonality of methane. Give us some detail about this new discovery.
Well, I thought I'd tell you right away that we've seen the seasonal variation, and we've tried to look at the data and come up with some explanations. And we've been able to rule out some of the sources. We don't actually think the meteoritic or delivery of interplanetary dust that can produce methane in the atmosphere, as Jen said, we don't think that's so important because we wouldn't expect to see a large seasonal variation. We might see 20%. Instead, we're seeing this massive change in the methane signal. And so what we consider, we look at the data, and the idea that best fits our data is the idea of subsurface storage. So way under the ground, under Mars, there's methane that's trapped. It may be trapped as clathrates or other materials. We don't know if that methane is ancient. We don't know if it's modern. It could be either. And we also don't know if that methane was created from water rock chemistry like serpentinization, or it was created by methanogen microbes. We cannot distinguish that. So, but the methane leaks or seeps up to the surface, we believe, and finds its way through cracks and fissures. And eventually, when it gets to the surface, we're then in a situation where the surface temperature can modulate or especially amplify the release of methane. So this is an exciting time because we have this seasonal cycle to constrain some of the theories and of the sources and sinks of this important gas.
Now, Chris, I want to talk to you a bit more about trying to distinguish between whether this is a biological or non-biological source of the methane. But to begin with, tell us about how you actually detected this. So in the case of Jen's work, they actually brought Martian soil into this oven and heated it up. But how was the methane variation detected?
Well, it's we're ingesting from the air, and if you look at Curiosity, around about waist height, there are two inlets, two little valves, and these valves allow the Martian atmosphere into our sample cell. Once it gets into SAM and into the tunable laser spectrometer in particular, we have tiny little infrared laser beam that multi-passes. It bounces 81 times between these mirrors, and that amplifies its sensitivity. So when you look at the laser light on the detector, you can scan the laser and look for the spectrum of methane. And the resolution, the spectral resolution is so high that we see individual methane lines, and in particular, we see a fingerprint of 3 lines together that can only be from methane. So our measurement is unambiguous. The signals we see, they come from methane and nothing else. And secondly, we measure them with high precision.
So let's go to the 2 different scenarios as to what would be causing the methane. I guess one is that we have a seasonal cycle because bacteria under the surface of Mars would be more active in the summer. Is that the idea?
That's a possible idea, but again, we cannot distinguish that from serpentinization, the reaction of water with olivine to produce serpentine and hydrogen. Once you get the hydrogen released in both in that reaction, it can be catalyzed either through metal catalyzed methane, that's the abiological route, or it can be catalyzed through enzymes in the microbes. In the cell potential.
So just sort of putting that into slightly simpler terms, if you have water that's interacting with volcanic minerals like olivine, you can also get similar methane that's trapped in the soil and preferentially comes up in the summer?
Yes, that's exactly right.
Right.
But we're hoping that we'll see we'll get more results in the future on this, especially from other missions. And as far as testing for the biological source or not, there are several more steps that can be made. We can look at the carbon-13 isotope ratio, for example, that could be suggestive of biological activity, and we can look at the accompaniment of other gases. So we have, and we can sit on the surface in Curiosity, and maybe one day we'll see a plume that's large enough where we can measure that carbon-13 ratio ourselves. So there's lots of exciting ways forward. But at this moment, again, the biological option is being held. It hasn't been ruled out.
So really exciting results about the methane and the seasonal variation of it on Mars. I want to take a moment just to say, for anybody who's been joining us, we're live at NASA. We're at the Goddard Space Flight Center, and we're also at NASA's Jet Propulsion Laboratory. And we'll be taking your questions soon live. So if you're on social media, use the hashtag AskNASA. And if you're joining us on Facebook, just put your questions in the comments, and we'll get to as many of those as we possibly can. Okay, so we've talked a bit about the latest results from the Mars Curiosity rover. One of the things that I just want to mention very briefly is that we have a mission to Mars on the way right now. May 5th of this year, we launched the InSight mission, and it will arrive at Mars November 26th of this coming year. And the exciting thing about the InSight mission is that it's looking at a different sort of definition of the word life. It's looking to see whether the planet Mars is still geologically alive. And by that we mean, are there things like Mars quakes? Is there any activity below the surface? How quickly is the interior of Mars losing heat through geological processes? So look for that. Coming this fall, we'll have another landing on Mars, a geological mission to actually see what the activity geologically is underneath the surface of Mars. Okay, well, before we go on, I do want to make one more trip back to the Mars Curiosity rover and to Ashwin. So, Ashwin, the amazing thing about this seasonal variation of methane that we were just talking about is you need to be there for many seasons. So tell us about the lifetime of the Curiosity rover, and you just give us a sense of what an amazing accomplishment this has been.
Sure. Yeah, you know, this is really wonderful to have this discovery that took so many years to not only take the measurements, but then to analyze as well. The measurements that Chris described come from an instrument called the Tunable Laser Spectrometer within the SAM instrument suite on Mars. And we've taken about 30 measurements with this instrument over the course of the mission. In fact, we're planning one on Mars today. So without having the rover survive for 6 Earth years, 3 Mars years, which is 3 times longer than its original mission, we would have never been able to discover these amazing seasonal patterns that are the heart of what Chris described. And finally, I just wanted to add a few more words to Jen's discoveries. One of the things that makes that so special is the role, how it fulfills the role that Curiosity has played within the Mars exploration program. NASA charged this mission with detecting organics on Mars, and we've certainly done that and expanded the range of organics that we know of today. But it was more than that, too. We also wanted to understand how organic molecules, organic matter on Mars that was incorporated in rocks 3 billion years ago would be there for us to survive, would be there for us to discover all those years later. What are the processes and environments that allowed it to resist that degradation? And that really is remarkable from Jen's results, that she's been able to determine what types of molecules and what types of environments allowed those molecules to persist to today. And this is not only important for Curiosity, as fulfilling one of our main mission objectives, but it's also important for future Mars missions who will seek direct signs of life. Missions like ExoMars and Mars 2020. And what this does is it gives us confidence that when those missions get to Mars, there still can be something for them to discover.
So Ashwin, you were just mentioning that the Martian year is a bit longer than the Earth year, and another thing is that the Martian day, tell us about how similar the Martian day is to an Earth day and how different it is. And I think you actually have some personal stories relating to this.
Well, yeah, there's an enormous number of similarities between Mars and Earth. They're very similar planets in some ways that are purely coincidental. Mars has a 24.5-hour day, and it has a tilt on its axis about 25 degrees, similar to Earth's tilt of 23 degrees, which makes the days and the seasons remarkably Earth-like. It also makes it incredibly interesting to operate a rover in the most efficient way on Mars, because the best way of doing that is to actually live on a 24 and 37-minute day, which all of us on the Mars Science Laboratory did for the first 90 days of the mission, before we all started going crazy.
You just come in about 40 minutes later every day. That would disrupt your sleep, I would think. Yeah.
That's right.
Somebody who's just joined us has asked us, where are you? Give us another sense about where you're standing and what sort of room you're in right now. Sure.
Yeah, we're at the Jet Propulsion Laboratory. We're in a place called the Mars Yard. It doesn't look like a yard in here, of course, because we're in a shed where the twin model of Curiosity lives. So behind us, you see the engineering model. Some people call her Maggie. And she's very much like the one that's on Mars, especially in terms of all the software on board and all the electronics. We have some of the instruments here as well. And this model can drive out into the yard, and we've used it for tests, including solving anomalies that occur on the surface of Mars. This is the model that was used to teach the rover on Mars how to drive and how to navigate. We can use it for a lot of things where we don't want to use the precious time and resources of the actual rover on Mars.
Well, but we're going to start by taking some questions now, but before we do that, I actually want to go back to Paul for just one more last thing. So, we've talked about landing this amazing rover on Mars, and then you have this incredible chemical laboratory on Mars. Tell us that story. How did we get this sensitive chemical laboratory on another planet?
Let me get to that question. You're asking an awful lot of questions. Let me have you throw one in my direction first, and
Okay.
that nobody will ask. And what did you do this morning, Paul?
What did you do this morning, Paul?
Thanks for asking. I thought you'd never ask. What we did this morning, actually, was look at data that came down from Curiosity that was deposited into SAM, and it's been months and months. So like Ashwin mentioned, we're just in the process of getting the drill back in order. We're just tremendously excited about that. So the search for organics is going to go on. But getting back to your other question, which is what it took to get the, the SAM instrument is just very complicated. It took a huge team of scientists and engineers and software programmers. It's an international team. We put this instrument together at NASA Goddard, but Chris Webster's team at Jet Propulsion Lab developed the tunable laser spectrometer. Our French colleagues developed the gas chromatograph mass spectrometer. In fact, this morning they were calling in from Toulouse because they were supporting operations of our instrument from Toulouse. So what we did was we spent several years putting this together. Assembled it at Goddard, tested it at Goddard. We put it in environmental chambers that exactly match the conditions we would have on the surface of Mars. We tested out the scripts, the software that we're going to run on Mars. And then eventually we brought it to Jet Propulsion Lab. And this image that you see shows SAM, that's that gold-coated box about the size of a microwave oven, being gently lowered into the rover. The rover is upside down at that point, and we lowered SAM into it, and then we did some more testing. We put the whole rover, in fact, in a big environmental chamber at JPL, and when we were sure everything was perfect, then the rover was ready to ship to the Cape, and everything got launched. So you see everybody there in bunny suits. The reason they're in those white suits is we did not want to bring our skin oils and everything else to Mars, because then Jen would have to report that we were finding things that looked like they came from Chris Webster or somebody else in the clean room at JPL. So it took a lot of work and great teamwork and several nations joining us. It's just been tremendously exciting.
Well, actually, what you were just talking about leads into one of the social media questions. So a person named 3D Physics from Twitter asks, how do you verify that the sensitive experiments haven't been compromised, either before reaching on Mars or after landing on Mars?
Yeah, what we do is we bring along standards, for example. We do what any good analytic chemist does in the lab. We run blanks. So for example, if we're looking to make sure that none of the residual organics in the instrument are producing the signals that we see, we go ahead and we run a full sequence of runs, but we don't put sample in the cup, and then we see what we got. So we work very, very hard to make sure that what we're seeing is from Mars, and we're very confident that, that what we're seeing is from Mars.
There's a lot of questions coming in right now. And as I mentioned, you're welcome to give us any questions that you have. You can either use the hashtag AskNASA, or if you're on Facebook, just simply put your questions in the comments. So there's several questions from many people, sorry that I'm not going to name you all, about can this methane that we're detecting ever be used as a fuel for future missions? Is it something that's useful for a human presence on Mars? Should we go to Chris for that? Or do you want to take a stab at that question, Chris?
Well, of course, there isn't, right now we're seeing low background levels. We're not seeing a lot of methane. Of course, methane is always useful as a fuel, but the ability to extract it from the atmosphere would not be efficient. But if there was a way one could access subsurface reservoirs, then it becomes a whole new ballgame. But that's really speculation at this point.
And I guess I'll, seeing as we're at JPL right now, I'll give another one to Ashwin. So we have a question about if you could have added just one more tool to the rover, what would it have been, right? Well, yeah, what was the thing that you really want on the rover that you just don't happen to have?
That's a tough one. There's probably a whole list of things we could add. I'm going to deflect and just say the next thing we want to do on Mars is exactly what we're doing with the Mars 2020 mission. Building on all the results from habitability and organics and the possibility of life that Curiosity has opened up, and looking ahead to having instruments on Mars that are built specifically to look for those signs of life.
So Jen, this seems like a good one for an astrobiologist. I mean, this is a difficult question. I mean, we have organic molecules. How do you know if they're processed by life? You know, what signs would you look for that might hint to you that this is something based on life?
Well, all life that we know of is based on organic molecules, even though we know that that's not the only way you can make them. So if we're going to look for life, the first thing you'd want to do is look for signatures associated with organic materials. And that was part of the reason why it's so significant that we found organic materials in the first place. It helps us guide where we're going to look for more of these. Now, in those signatures, they might be things about the chemistry of the organic materials, or perhaps there are things about how the organic materials are stuck together in shapes, how they're packaged. Maybe it's going to be about their isotopes. There's a whole suite of chemical and physical things that we can measure that might indicate signatures of life.
Let me jump in there. I mean, Jen herself went up to Svalbard one time to one of the farthest, most northern springs, a hot spring way up in that cold area. And then we put some of this material in the gas chromatograph mass spectrometer, and these patterns showed up that show the length of the carbon chains, and bingo, that was the signature for life. So those are one of the many types of things we'll look for.
That's right, and there happens to be another experiment on SAM that we haven't used yet. It's one of the wet chemistry experiments. And if those types of things are left behind in the organic material that we come across, assuming we will come across more, perhaps, if it's the right sample and the right compounds are there, maybe we'll get some hints.
So I understand we actually have a media question that they're going to try to feed in on audio. So if I could ask for that question, we're going to try to have an audio feed at the moment.
Thank you. Our first question is from Chris Van Cleve from CBS. Your line is open.
Go ahead, ask your question, please.
Thanks for doing the call here. And forgive me if I missed this at the very beginning, but if you could sort of translate this into terms my grandmother would understand, why is methane so exciting, and what is it that you found that is so intriguing about this organic chemical, or compound, molecule that's different than what we kind of already knew, that there were already the building blocks of life potentially on Mars?
Okay, should we go to Chris for the methane question? Did you hear that, Chris?
Yes, I would start right away by saying whenever you have an oxidizing atmosphere and you see reduced compounds like methane or hydrogen sulfide, they're always very exciting. And in particular for the Mars methane, because 95% of the methane on Earth is produced from biology, it's produced from termites or rice paddies or cows or sheep or termites, etc. Because of that, there's always been this interest in Mars methane as its potential biological origin. And that's true also because we know it only can last for 300 years. So if we see methane in the Martian atmosphere, that means something is happening today. It's being released or it's being created. And I'll hand over to Paul to answer the second part.
Well, let me add to that. I mean, on Earth, like Chris mentioned, most of the methane in the atmosphere comes from biology, from rice paddies, or from a lot of different processes. But most of the oxygen on Earth also comes from biology. Billions of years ago, the cyanobacteria got serious, and they started cranking away. And so in our atmosphere, we have both a combination of methane and oxygen. And that's what some of the folks who are building these big telescopes, follow-on telescopes to the James Webb, for example, that are going to be out in space looking at exoplanets, they're going to be looking for combinations of oxidizing and reducing things that might be evidence of life. So it's really fundamental, it's really interesting, and I sure hope Grandma understood that.
And I understand we have another question from the media that'll come over audio.
Thank you. Next question is Nancy Atkinson, Speaker.com. Your line is open.
Hi. There was a similar announcement of detection of both methane and organic materials back in 2014, also by the Curiosity rover. How is this announcement different, or how have you built on the findings from 2014 for this detection?
I know we were just talking about that beforehand, as that being a very good question to ask. So I think, Jen, you want to take that?
Sure. In 2014, we reported the discovery of chlorinated molecules. Now, these are carbon structures that have a chlorine attached to them, and there was a few of them. It was a significant discovery. It was the first time we were able to confirm organic molecules on Mars. It was the first time. And we've been looking for this for a very long time, with the hopes that we would find it with the Viking instruments in 1976, and finally with Curiosity in landing on a lake bed, that we could actually discover these. Now, we were left with, what do these mean? The thing about the chlorinated molecules is that it's not what you typically find in natural samples. And so we weren't sure what the significance was at the time. However, it did give us a lot of motivation to keep looking, There have to be other layers in the Gale crater, other layers of the lake sediments, and we wanted to find more organic molecules. And that's exactly what happened. We drove 4 miles away. We started down at the lowest part of the crater. We drove 4 miles away and got to the base of the mountain, and the first layers that we looked at contained all of this organic material that we're reporting today. So we have essentially expanded upon the inventory of the organic molecules, and now we have a better sense of how they were preserved in the rocks in the first place.
So should mention that your report, Jen, showed significantly greater abundance than we had detected before. And one more thing to add is, in almost every experiment we do, as we heat up the sample in this little quartz cup, we see a big oxygen signal, and the oxygen signal comes from a mineral that breaks apart, that contains chlorine and oxygen. But then the hot oxygen can react with some of the organics and produce carbon dioxide. That's called combustion. And what Jen did with part of her analysis was really to look at the high temperature release of organics that might have escaped some of that destruction. But there's still things there that we're not seeing. So I mean, that's really motivating our desire to get back and with different techniques, really look for a more comprehensive suite of organics, try and figure out where they really came from.
Right. And Chris, would you like to continue your answer as to why what we're announcing today is different from what was detected before as far as the seasonality of the methane?
Yes, it's very different, because in the past we were seeing spikes that we couldn't understand, and we had ideas about them, but we couldn't constrain them because they only occurred occasionally. So now first of all, we've measured the background level over 2 years, so we know the total amount of methane in the atmosphere is constrained by that. It's a low average value of only 0.4 parts per billion. It changes a lot, but the total amount is low, so that also constrains the magnitude of pulses and spikes elsewhere that would mix into the atmosphere. So this is a very important observation. The fact that it changes so dramatically, this factor of 3, has got to be duplicated by models that explain it. Now, we do think that the spikes and the low background seasonal cycle are related. It's quite conceivable that the same reservoirs that are slowly seeping methane up to the surface are also occasionally causing large pulses of the gas to be released.
Well, thank you. And I understand we have another media question. So we'll go to the audio line now.
Thank you. Our next question from Leo Enright with Irish Television. Your line is open.
Thanks very much. If I could just summarize, as I understand it, the takeaway is confirmation of a seasonal cycle of methane, and also, quite noticeably, a detection of thiophene in the subsoil. So if I could ask, what follow-up investigations, specifically from the Europeans, could support and add to this? Specifically, the trace gas orbiter presumably can do the kind of isotopic analysis that you can't. So can you foresee that being able to contribute? And secondly, still with ExoMars, but in the future, they're talking about drilling to 2 meters. Is the gas chromatograph on ExoMars the same as the one on MSL? And can it do the work that you say needs to be done?
Let me take a first shot at that, and Chris may want to jump in. First of all, let me say, Leo, it's great to hear somebody saying methane instead of methane, like Chris Webster does all the time. But starting with the ExoMars mission, we're actually very proud of the work that our team did here at Goddard to deliver a mass spectrometer to an experiment on the ExoMars Rover, which is planned to launch in 2020. That instrument is called MoMA. And we greatly expand the techniques that we use there to look at these samples that come up from 2 meters below the surface. The first thing we do is totally different. It's never been done before on the surface of another planet. We use a laser, basically, to put a pulse of energy into the sample that's in front of the instrument, and then we let some of those ions up into a mass spectrometer. It has a very high mass range. We can do things like find a high mass peak. It's a very soft ionization technique. And we can do what's called tandem mass spectrometry. We can break that molecule apart into its constituents and really try and get at the structure. It's a very powerful technique. We also have a GCMS, a gas chromatograph mass spectrometer, and that also was a contribution from our French colleagues. They're really good at understanding how to do that separation of gases that Jen talked about. And they do the derivatization chemistry, the chemistry that tries to pull out interesting things like amino acids a little bit differently. We kind of have a lot of fluid in a cup. We puncture the cup and put sample in. And they try and control that a little bit better by having basically a little container that'll break apart at exactly the right temperature, put the derivatization in, and then do separation. The technologies have also advanced a bit. For example, one thing we'd be very interested in finding is chirality, the left and right-handedness of an amino acid, if we're lucky enough to find an amino acid. On life, they're not equivalent, but in meteorites, they're largely equivalent. So with MoMA, we're trying to solve that problem as well. So technology advances and our ability to think through what we can put in place on another planet advances. And maybe Chris should talk about what the European Trace Gas Orbiter, which is also looking for methane, what they hope to find. I understand that's just getting going in operation around Mars, and they've not been doing much reporting yet, but they're just getting their initial set of data.
Sure. A comment, Chris?
Yes, the exciting thing about TGO, as it's called, it's going there to look at a lot of gases in the atmosphere, but especially methane in particular. As Paul said, they've started to make measurements, so we're all waiting with bated breath to see what they find. But they're going to, in particular, by mapping it globally, they'll be able to relate their measurements to ours. Now, one thing that they could do is if they see large plumes somewhere, even though they don't have a lot of spatial resolution, they could potentially pinpoint or hone in on an area of Mars that seems to be associated with the increase from the surface. That would be a very exciting find because it tells us where we need to go to get the better signals when we look at it. And that's important. You asked about the carbon-13 isotope ratio. Remember, there's 100 times less carbon-13 methane than there is regular methane. And if you want to measure that to 1%, you're stuck with this extremely high sensitivity needed. So if the Trace Gas Orbiter sees significant amounts of methane, they will certainly be able to make that measurement. But if they see mainly the low background levels that we are seeing of 0.4 parts per billion, it's going to be a challenge.
So those are, if you don't mind, we have a limited time for questions, and we've had some pretty technical answers. I actually want to go to some of the middle school students that are actually live tweeting us right now. So apparently I need to call out Mr. Soldy's aerospace class. So you guys are sending us lots of questions. So they're asking us about the implications. Does this discovery have any implications for the future colonization of Mars or human presence on Mars?
Sure, it could. When we have organic materials in the rocks that we found in Gale Crater, there might be wider spread. And perhaps those would be a resource for humans. We don't know what that would be exactly. Perhaps there are organisms there that we don't know about yet. They might be helpful for farming. There might be organic materials that could be used as fuel for both farming. Organisms, including plants, need carbon in order to turn it into their own biomass. And perhaps there's other ways of using it to generate fuels that people there would actually use. Those have not been fully investigated yet, but I know it's an open book on what technologies we might use in the future on Mars.
And actually, there's sort of a follow-on question to this. So there's Mac from Pier Middle School who asked, would it be possible to bring samples back from Mars?
That's a great question, and Ashwin actually is the perfect person to ask that because there's a program at Jet Propulsion Laboratory that's doing exactly that.
So Ashwin, is it possible we might someday get samples back from Mars?
Yeah, we sure hope so. One of the things that we're working on here at JPL is the mission that I referred to earlier called the Mars 2020 rover. It has some really sophisticated instruments that it's bringing with it to Mars to do these kinds of investigations, to look directly for signs of life. But it also is proposed to bring samples back to Earth one day. It requires a few follow-on missions to do that, something to land a rocket on Mars, and then to gather those samples from 2020 or where 2020 has left them on the surface, to bring them back to that rocket, and then to blast off from Mars, and then to rendezvous with the spacecraft that we've now put in Mars orbit and bring it back to Earth. That's a lot of really actually cool technology that JPL has to invent in the next couple decades to accomplish that.
And this is actually something that kind of leads on to that. We are planning to return to Mars. We have an active Mars exploration program, and one of the things we're trying to do is figure out where the best places to land next would be. So there's some questions here about how are we determining the best places to, where do we explore next on Mars?
One of the big questions is where else are we going to find the organic carbon? And there are various environments that we understand on Earth as being good places that we can collect organic materials, that get preserved, and it's also good places for life. So for instance, a lake bed, like what we found in Gale Crater, is a perfect example. But there are others, hydrothermal systems, where hot fluids and gases coming out of the ground can provide a lot of chemicals that organisms can use. We see these in the ocean on Earth as hydrothermal vents. There are records of rocks on Mars that suggest there are ancient hydrothermal systems. So that might be another place for us to look. So right now, the larger Mars science community is talking about where it is they want to go. And these are some of the key scientific motivators for choosing sites.
Let me add to that a little bit. We have two things. We have a huge amount of data because we have very capable orbiters orbiting Mars. And on those orbiters, we have imagers that can see a few feet. We can see our rovers on the surface of Mars. We can see the rover tracks. So that's one tool we use. The other tool we use from space is spectroscopy. One thing that brought us to Gale Crater was finding minerals that were hydrated, that had water. So that was a clue there might have been a lake there. But the other really fun thing about Mars is on Earth it's very, very hard to find old rocks. And the reason is Earth is so active still. You see the Hawaii volcanoes going off. And so the landscape keeps changing. Plate tectonics it's called. It moves the continents around. On Mars, that stuff all froze out billions of years ago. So, half the surface practically is very, very, very old. So we can get at the ancient history of Mars by landing on the surface and looking at what happened early in the history of the solar system.
That's right. The modern landscape is actually a very ancient one.
Yep.
Yeah.
So we're going to go to another media question. So this will come over the audio line.
Thank you. Next question is from Mike Wall with Space.com. Your line is open.
Thank you, guys. Really appreciate your time, and this is all very interesting stuff. This is probably a question for Ashwin, but if anybody else wants to jump in. But you're coming up on 6 years now on Mars, and so how do you think the results and totality of what Curiosity has found will sort of shape its legacy? And I know it's weird to talk about something because it's still really strong, but it's almost been 6 years now. So yeah, how do these fit in with the overall mission and how it's going to be remembered and what its legacy is going to shape up to be, do you think?
And Ashwin, I hope that came through well. I believe the person's asking about how these discoveries will fit in with the legacy of the Mars Curiosity rover and your view of that. Some comments from you?
Yeah, it's a great question, and sometimes being involved so deeply in the day-to-day sort of forget to look at the bigger picture. But as I mentioned earlier, the way I think about it is really that we have a role in this ongoing exploration of Mars that's now been going on for more than 50 years and will continue in the future as we get closer and closer to understand if life ever originated on Mars and then eventually sending humans there and maybe even colonizing Mars one day. And the role that Curiosity was designed to play was to figure out if Mars ever was habitable. We didn't know that before Curiosity went there. We didn't know that lakes survived on Mars in this time period from 3 to 4 billion years ago, that they were surviving on Mars for up to millions of years. And we didn't know the extent that organic matter was present on Mars, could be concentrated in these environments, and would be preserved for us to study today. So those are some really big questions that the mission was specifically designed to address. And with these results, I couldn't be happier in how we've been able to meet those objectives and provide them as material for future missions to use when they're designing where to go and how to accomplish their science.
I think we just have a little bit of time left, but I think there's one more question from the media. We're going to try to sneak in, so please go ahead.
Thank you. Our next question is from Hillary Breck, Business Insider. Your line is open.
Hi, yeah, thanks for taking my question. I just wanted to ask a question about those class rates that the methane is coming from. Do you think that it's the rising summer temperatures that are warming those up and that's how they're releasing it?
Chris, want to talk about some of the possible sources for the rising methane levels in the summer?
Yes, you asked about clathrates. So there is clathrates are stable on Mars. They're created through the high pressure, of course, initially. They could be a source, but we don't believe, remember, once you get under the surface by several meters and below that, you don't really have a seasonal cycle there. It's pretty constant temperature. So the clathrates or other reservoirs might be leaking or seeping the methane continuously throughout the year. It's only when it gets to the surface, where the surface temperature has that seasonal cycle, that modulates the release of that methane into the atmosphere. I think that was your question.
Well, that's actually all the time we have. If you submitted questions via social media that we didn't get to, we're going to spend the next hours and days trying to get to as many of them as we can and answering them online. But for now, thank you so much for joining us. We were live at NASA, and have a great rest of the day. Bye.
We want to know, has life ever existed on Mars? Do pockets of life persist on Mars today? NASA's approach to answering these questions is to break them down into smaller and smaller steps. First, we need to know if ancient Mars was habitable. Did it once have the right climate and the right chemistry to support life? The Curiosity rover is investigating these questions by looking for organic molecules containing carbon. Organic molecules are the backbone of all life on Earth, though they can also come from non-living sources. Today, the surface of Mars readily destroys organics, making them difficult to detect.
We are decelerating.
6 years ago, Curiosity landed in Gale Crater on an ancient lake bed. A few months after arrival, it drilled into sedimentary rocks and detected traces of organic molecules using an instrument called SAM. Now Curiosity is climbing the mound in the middle of Gale Crater, and SAM has made a subsequent detection of organics. This new detection is exciting because it comes from rocks that are billions of years old. That means that the organic material within them is extremely ancient. Some of the organics that SAM has detected contain sulfur, likely introduced through geological processes. Sulfur can act as a preservative, binding organic molecules together to make them tougher and protecting them from oxidation. In fact, sulfur is the element that makes hair and fingernails tough, as well as vulcanized rubber. Martian sulfur has probably had a similar effect on these old organic molecules, helping to preserve them over geological timescales. SAM made the new detections by heating samples of crushed rock to very high temperatures, above 1,000 degrees Fahrenheit. This vaporized the samples and released several species of small hydrocarbons like benzene and propane. Because the hydrocarbons were released at such high temperatures, they may be the fragments of bigger, heavier molecules within the rock similar to kerogens. On Earth, kerogens are found in rocks like black shale and coal and are the products of ancient plant and bacteria. We don't know if the recently discovered organics on Mars are of biological origin, but it's exciting to find such old material preserved right at the surface. This finding is also encouraging for future exploration. NASA and the European Space Agency are preparing to send the next generation of rovers to Mars in 2020, carrying new technologies to search for signs of microbial life. In the distant past, Mars was much warmer and wetter than it is today. The rocks at Gale Crater tell us it was once an environment where life as we know it could have survived. The discovery of ancient organic molecules shows that another ingredient of life was present at that time, and it broadens our understanding of habitability of both ancient and modern Mars.